Thank you. Right, so I've got 40 minutes to, my brief is to explain the principles of imprinting, the principles of imprinting disorders, and just to mention the new clinical consensus on one of the classical imprinting disorders, which is called pseudohypoparathyroidism. So I'm going to be able to tell you in 40 minutes that all of this is very, very easy to understand, and I think that's very important. So, to start off with, the principles of imprinting are very easy to understand, and I'm sure you all know this. So the point about, obviously I don't have to say this in a genetics session, in the ESHG, but we are all genetic creatures. Basically every single cell in our bodies contains exactly the same DNA, but obviously they're not all doing the same things with it. So we have, every cell contains DNA sequence. This DNA is organized into genes which are present, being transcribed in different directions, in different ways around the genome. And the genetic structure of the DNA and the non-coding DNA coils it into certain structures which dictate its expression. And that expression is therefore, has to be marked upon the genes. And the particular organization of the genes in any given cell at any given time will determine which genes are transcribed, and which genes are transcribed determines what the cells look like. This much is obvious. Oh, I'm sorry, I have a few little problems with my presentation. This is a Mac to PC thing, so I will do my slides if I have to in interpretative dance. Don't worry about it. So, DNA describes proteins and RNA, and the epigenetic marks on the DNA, the non-coding epigenetic marks, determine when and where genes are expressed. Now, most epigenetic marks are diverse and dynamic. I'm not going to go into the vast majority of them because they are so complicated that they are currently verging on, with a few honourable exceptions, too difficult to interpret for clinical actionable situations. I'm going to focus on DNA methylation, which is a durable and detectable epigenetic mark which is put on cytosines within CpG residues in DNA. This is relatively easy to identify, and it's durable to normal DNA preparation mechanisms, obviously. So basically, to put it much too simply, DNA methylation on the promoters of genes renders them less likely to be expressed. So DNA methylation is inversely proportional to gene expression. And so you will see me using what I call lollipop notation. It's very standard. Here we go. A filled lollipop indicates a methylated cytosine in a CG dinucleotide. An open lollipop signifies an unmethylated cytosine. So basically, a lack of methylation on a promoter is more likely to be associated with active genes, whereas DNA methylation is more likely to be associated with inactivity. So that's the basic principle of DNA methylation, which is a very detectable mark on DNA. Right, now, so what I've been trying to say so far, simply, is that if the genes that a cell is expressing reflect what its phenotype is, and that's controlled by epigenetics, to say it the other way around, the epigenetic marks on DNA indicate what the cell is doing, and they indicate what age it is, where it's come from, what tissue it's in. Therefore, in principle, epigenetics is the developmental status of the cell written on its genome. I hope that makes sense. Now, the most remarkable, the most distinctive phase at which this is true is during reproduction, because what we've got here, you have to have a certain number of cells from these highly differentiated, so we're terminally differentiated somatic structures, but just a few cells from each of us will take on the role of becoming germ cells, and they're going to have to take away all their epigenetic marks, and they're going to have to form this new thing which is completely totipotent, and it goes through the stage of going from 50 trillion cells, which is us, down to one cell, which is a new individual, and then out from there, beginning the process of differentiation to become a new individual. So basically, all the epigenetic marks of a terminally or a very complex differentiated individual have to be taken down to their bare minimum, which is one new cell. That's why epigenetics in reproduction is so interesting. So, what we have to do is we have to clear the epigenetic marks of the differentiated person during development. Otherwise, of course, you can't become a new person. So, global epigenetic reprogramming occurs twice during reproduction. The first thing is that let's get rid of that. The germ cells, the cells that become the germ cells of the new individual, must wipe basically all epigenetic marking. So these cells have a very nearly completely clean epigenetic slate. They can be anything at all. Then, however, of course, what happens is these germ cells become two of the most highly differentiated cells that actually exist in the whole organism. They're both haploid. One is absolutely enormous and the other one is completely tiny. They're really, really divergent, highly differentiated cells, yeah? And that's because they've got this completely different epigenetic reprogramming. Then, of course, there's fertilization, and we have the combination of the two genomes. So we had the sperm genome that's got its own particular pattern of gene expression, its own epigenetic marks, the egg that's got its own epigenetic marks, its own transcription. In the zygote, these two genomes come together. They are not the same. They're two haploid genomes, they've got different marks on them, they're both from two differentiated cells. Therefore, the first thing the zygote is going to have to do is wipe it again. This second post-zygotic wiping and resetting is not so extensive as the one in the germline, but it's pretty extensive, okay? And then what's going to happen, as you get down to the blastocyst stage, you approach a minimum, again, of epigenetic marking, and then from there on, you're going to start to have the differentiation processes, which will lead to the new organism. Now, this thing keeps kind of moving on when I'm not expecting it to. Oh, sorry, I apologise for that. Right, so now a small number of genes evade the second wave of reprogramming. So what happens is a small number of genes in the germ cells. The whole genome is obviously, it has epigenetic wiping and resetting. Some genes will be expressed in the sperm and not in the egg, or vice versa. And then in the zygote, these two are recombined, the two genomes are recombined, and then some epigenetic marks during the zygotic wave of reprogramming evade both the deletion, the wiping, and any subsequent resetting. And they simply don't change thereafter. That means that certain genes with these epigenetic marks on retain a memory of the parent they came from. And it's not in the DNA structure itself, it's on the DNA in the form of epigenetic marks. And most specifically for me, most clearly, in the form of differential methylation. So it's simply that DNA methylation didn't get wiped post-zygotically. Right, so what do we therefore know about imprinting? This is the second part of my talk. Now, genomic imprinting. So what I've got here, I'm just going to show you what I'm representing. Here's my target individual who has got a DNA methylation mark that is specific to one parent, and it's one that's on the father. The father also is going to have that specific methylation mark that he will have had from his father. The mother has that mark that she will have got from her father. So, imprinting marks, these special marks that don't get changed in zygotic reprogramming, first thing, they're uncommon. Imprinting affects a few hundred genes because there are very few of these marks that actually evade zygotic reprogramming. Secondly, they're present in nearly all cells in the organism. There are a few exceptions, but I'm not going to go into that. Thirdly, they're present permanently. And this makes imprinting marks, unlike most other epigenetic marks, tractable. Because I can take a blood sample or a skin sample or any other sample from the individual, and I'm likely to be able to read the imprinting that's in the entire organism. And that makes them relatively easy to use for me, and obviously easy to use diagnostically. Imprinted loci, these parts of the genome, they're numerous. Whoops, sorry, I keep doing that. Sorry about that. They're distributed across the genome. There we are. So I've just put on stars on here, indicating these imprints. These are methylation marks that derive from mother. The blue ones are imprinting marks deriving from father. They're not all currently associated with classical imprinting disorders. So what I mean to say is that, so far as we know, the gene dosage or the gene quality of most of these genes is actually not associated with any syndrome, either because we haven't discovered it yet, it's not compatible with life, or it has no phenotypic effect. We have yet to discover that. Right, next point. Imprinted genes are hemizygously expressed, at least the ones we're thinking about. They're expressed from one parent only. The critical ones that we're thinking of in terms of imprinting disorders are tightly regulated in growth and development. And this means that if the level of expression is important, it means that altering that level of expression may have deleterious effects. And that's why some imprinted genes are associated with imprinting disorders. So far, so good. So we have, for example, paternally expressed genes here, maternally expressed genes, and the balance between them is critical for optimal growth and development. Now, what this means is that imprinting disorders are caused by changing the expression level of imprinted genes. And there are three different ways of doing this, basically. Firstly, you can alter the gene itself. There can be genetic or coding errors in the one allele that is being expressed. So if we imagine our paternal allele that should be expressed, if it's deleted or duplicated or mutated, then there is effectively no expression. That is the consequence of hemizygous expression. This is obviously heritable, but dependent on the parent from which it's inherited. Secondly, there can be a reproductive error. Uniparental disomy means inheriting, as it were, apparently inheriting both copies of a chromosome from one parent with no representation of the other parent. This, in principle, is not heritable unless there's an underlying chromosomal disorder. And lastly, there can be an epigenetic error. That is to say, there's no apparent underlying problem at all with the DNA or with the chromosomes, but simply one gene has acquired an aberrant imprinting mark. Now, in principle, these aren't heritable, but I'm going to go into that. Okay? So those are the three different ways that one can get a change in imprinted gene expression. And if that imprinted gene is important, an imprinting disorder results. Now, what are the practical challenges of looking at imprinting disorders at the moment? Imprinting disorders are epigenetic syndromes. That is to say, there's not necessarily anything you can see on the DNA, but they affect genetic gene use, and they affect growth, development, metabolism, and behaviour. The cumulative prevalence of these disorders is greater than 1 in 5,000. Most of these are very, very clear clinical gestalts, clinical sets of features that are very recognisable and lead to a clinical diagnosis of an imprinting syndrome. The disorders have, I'm just going to pause and say hello to some of these. So chromosome 11, there are two imprinted loci together on chromosome 11. They cause effectively opposing imprinting disorders. One is associated with overgrowth, particularly of certain tissues. One is associated with growth restriction. Frederick is going to speak very much more about these, and so I'm going to leave these aside. Chromosome 15, the UBE3A locus on chromosome 15 is associated obviously with both Prader-Willi and Angelman syndromes. So what we do have is evidence that some of these loci are so sensitively regulated that changes in expression either way can result in imprinting syndromes. I just want to offer you, also I have here chromosome 6 and chromosome 20. Sorry for the picture that's gone awry again. Chromosome 6 has hitherto been associated with one imprinting disorder, which is transient neonatal diabetes, caused by overexpression of genes of an imprinted locus. There is now evidence that maternal uniparental disomy of chromosome 6 also is associated with the syndrome. Likewise, chromosome 20, where effective paternalization of imprinting causes pseudohypoparathyroidism, there is now evidence that maternalization on chromosome 20 causes a growth restriction syndrome. So that's the range of recognisable disorders at the moment, broadly. The point about these is that so they have heterogeneous clinical features. That is to say, not all children with one of these disorders will have exactly the same clinical features. They have overlapping clinical features, and that includes things like growth restriction and certain kinds of dysmorphism, and that's part of the challenge. I'm going to go into that. One of the critical features is Mosaicism. Because these disorders affect the post-zygotic reorganization of DNA methylation, an imprinting disorder need not necessarily affect all cells in the individual. So an imprinting disorder can be mosaic, affecting some cells and not others. That has its own challenges. The last thing I'm going to talk about is that some imprinting disorders don't affect just one locus or one imprinted gene, they affect several. So I'm going to come on to that. Right, so firstly, one of the challenges of imprinting disorders is heterogeneous and overlapping clinical features. What I've got here is three. So this is effectively, I'm thinking about Silva-Russell syndrome, which is, it has critical features including, I'm not actually going to touch the screen because every time I do something moves, so you can just read it basically. But you can see that the critical features of Silva-Russell syndrome are a reduced birth weight, relative macrocephaly, postnatal growth restriction, feeding difficulties, asymmetry, these kinds of features. Now actually, not all children with Silva-Russell syndrome have all these features, and there's a sort of cumulative clinical score that needs to develop. But also, depending on the genetic etiology of the syndrome, they have different proportions of features. So for example, let me see. So Silva-Russell caused by UPD7, maternal UPD7, as you can see, isn't always associated with postnatal growth restriction. It's certainly very rarely associated with asymmetry because it's a, there it goes, never mind. Right, Temple syndrome, and I'm very glad to say Masayo Kagami is here and she's presenting a poster on Temple syndrome. It actually affects chromosome 14, but it has related clinical features. And more recently, it's also been shown that maternal uniparental disomy of chromosome 20 has also a growth restriction phenotype attached to it, but again with slightly different clinical features. But not all children with these genetic etiologies have these features. What I'm saying here is that if you see a child whose clinical features are consistent with an imprinting syndrome, it may actually be consistent with multiple imprinting syndromes. And that's what we're seeing, is that the molecular etiology is an important contributor to the overall diagnosis, just as the clinical etiology is, the clinical features are. This is effectively the molecular features are altering our perception of the diagnosis. Just to mention another example of mosaicism, and this I'm just going to touch on chromosome 15 because Fred's going to focus on 11 when he does his talk. So I just wanted to mention again, the molecular etiology of Angelman syndrome patients actually influences their features, very particularly if you look at the children with, let me see, for example, having no words. The most prevalent cause of Angelman syndrome is a deletion of the imprinted region, and most of these children are completely non-verbal. But actually, if they've got different etiologies, they can have some words. And likewise, one of the other points is that Angelman syndrome is not associated with obesity, with changes in body mass index. And that's because the major genetic etiology doesn't have a high body mass index, but some of the others do, actually. And most interestingly, mosaic imprinting errors at the Angelman syndrome locus are becoming more and more recognised, but they haven't been until now because they actually don't have very similar clinical features. What you can see is that they really can have a raised BMI, and they normally do have some words. So molecularly, they've got Angelman syndrome. Clinically, they look quite different. And so this is something one has to be prepared for. Now, the next thing I just want to move on to is MLID, multi-locus imprinting disorder. Some imprinting disorders affect not one locus, but multiple genes. Now, the involvement of multiple loci suggests that something is going wrong with the entire process of epigenetic resetting postzygotically. A lot of children with MLID have mosaic features. That is to say, they don't have a given defect is not present in all cells of the organism, all cells of the child, sorry, as it were, and the different imprinting disorders are actually differently mosaic, so some may be more affected than others. This suggests that it's happening in a patchwork way, in a mosaic way, early in development. Thirdly, MLID is epigenetically heterogeneous, so there aren't very clear patterns, I'll show you this, and clinically heterogeneous. Now, because until now, the imprinting disorders have been classical descriptions, this means that MLID has basically not been described until more recently. And frankly, they may be out there and we're not seeing them because we don't recognise them as having an imprinting disorder at all. Fourthly, MLID can be found in siblings. And this suggests that, at least in some cases, it's frankly genetic. And I'm going to show you two classes of mutations. Right, so firstly, there's this gene ZFP57. So ZFP57, it's a DNA binding factor, it's a transcription factor. And simply what it does is it associates with certain other genes early in development and it guides them to certain hexamer sequences on certain parts of the DNA. And what this means is that because it's got a specific DNA binding site, actually, if you see children with recessive mutations of ZFP57, certain loci are affected. And this gives them certain clinical presentations, and it's really quite distinctive. And so I have never seen a child with ZFP57 mutation that didn't have a particular phenotype and a particular epigenotype. Still could be ascertainment bias, I admit. Now what I've got here, I'm sorry, it kind of looks a bit hair-raising. I've got some of the classical and some of the other imprinted loci that I would tend to look at using targeted diagnosis. A white box means unaffected. A blue box means it's got a methylation change, okay? And what I've got, effectively each row is a child that I've been looking at. And so I direct your attention to the bottom of the red box. ZFP57 children, I think you can see this, I'll try not to trigger this. There we go. These children have very specific patterns of epigenetic change affecting 3 loci that actually aren't very highly affected in other disorders. And if you see that pattern, you're likely to see such and such a phenotype, which is neonatal diabetes with. That's what that is. Now, we've got a whole load of other children who've got multilocus imprinting disorders. In fact, quite a significant proportion of children, if they've got an imprinting disorder with an epigenetic change, they're quite likely to have more than one other. And the better our techniques get for measuring this, the more clearly we see this. So they've got heterogeneous epigenotypes, they've got heterogeneous presentations. If you look down the left-hand side here, we've got Silva-Russell syndrome, Beckwith-Wiedemann syndrome, transient neonatal diabetes. They can have all manner of things. And we don't really understand why they've got this. So why do these children have these heterogeneous presentations? Now, honestly, we're doing our best. And so I think several labs across Europe and across the world have been looking at some of these children. And I just want to cite 3 genes, just mention 3 genes that we do find to be affected in some of these individuals. And these are all NLRP genes. And these have been shown to have mutations or variants in a subset of children with MLID. What you can see is that if these variants are not associated with a particular epigenotype or a particular presentation, this has made them quite challenging to identify. I just want to explain why, what I think they might be doing early in development. Okay, so effectively I'm just going to answer these questions. Why are these children different? Why are these genes different? When and how do they act? Okay. So what I want to do is direct your attention to when this second phase of epigenetic resetting is taking place. What we effectively have is an oocyte being released from the ovary. It's going to be fertilized somewhere in the fallopian tube, and then it's got to undergo its first few cell divisions before it's going to implant in the uterus. Okay, so it's kind of out there on its own. So just firstly, I'm just going to think about the DNA methylation in the zygote during that time. So the maternal DNA methylation rises. Let's see if I can use this. Maternal DNA methylation rises in the developing oocyte and then is gradually lost broadly by reductive. So effectively each DNA replication, methylation is not replaced, and so gradually the mother's DNA is demethylated during this time. And then after about blastular time it starts to go up again. The father's DNA, the sperm DNA methylation, is set in the sperm, and then pretty much at the time of fertilization, within the first cell cycle, it's all removed. There we go, and then it sort of varies, just a little bit more is lost through reductive, replicative loss. Then imprinted loci are meant to be completely impervious to this. Now I'm going to think about DNA transcription during this time, because this is interesting and it's not terribly widely known. So firstly, the oocyte DNA transcription, the oocyte during its growing phase accumulates huge quantities of RNA and proteins that it's going to need for doing this first few cell cycles here. And then DNA transcription actually, so the level of DNA transcript declines. And that's because, in fact, there is no new transcription until about the 8-cell stage. So the zygote doesn't actually transcribe anything until it reaches about the 8-cell stage, which is quite interesting. That means that effectively whatever is in the oocyte here, this unique pattern of transcription and translation products here, means that the DNA is actually, it doesn't support the retention of DNA methylation. So in fact, among the things that are being lost here is stuff like DNA methyltransferase, UHRF1, things that kind of guide DNA methylation to where it should be. And the decline in these factors means that DNA methylation is lost during this time. So what I want to suggest is that actually, effectively, we have an egg timer. The fitness of the egg, the fitness of the zygote, the age of the egg, the age of the zygote sets the quantity of protein and RNA it's going to have in it, the quality of RNA and protein it's going to have in it. And that is going to influence what it can actually get up to. So what's in here? What's in here in the egg is going to influence what gets here into the blastocyst. And that's why I think that some of these maternal effect, or these genes that are present in the oocyte, coded by the mother, are so important because they help to organize some of these transcripts. I'm not going to go into this because it's, there we go. So I think that during its early development, most eggs they have a decline in their normal DNA, their gene transcription, and then it's going to start again once it gets to the blastocyst stage. But if there's a problem with the egg, either because it's old or tired or there's been an environmental insult, or because there's a genetic susceptibility, there is going to be a significant problem with that egg, and it is not going to develop right. And if it'll either just die, and you have effective infertility, at least from that oocyte, or you might have a crisis. And if there's a crisis, you may have an imprinting disorder. So that's why I think that MLID is partly genetic and partly not genetic. So I think that some multilocus imprinting disorders have maternal effect mutations. The consistent phenotype is MLID. Nothing else is consistent. That's why they're hard to find. I think these things have a spectrum of reproductive outcomes. So what I reckon is that maternal effect mutations that compromise early development can disturb epigenetic marking. This can include maintenance of imprinting. If you disturb maintenance of imprinting, you can get variable phenotypes. Maternal effect variants and non-genetic factors can work together. So there can be either a really, really serious genetic predisposition to an imprinting disorder, or it could just be there's been a terrible environmental insult, or both. And that's one of the reasons I think this is quite hard to see. What I'm saying is that I think that actually epigenetic imprinting disorders is nothing else, in many cases, than a relic of something that went wrong in early development. And in some cases there's an epigenetic predisposition, in some cases there's a genetic one, and in some cases there's a mixture. So that's why they're relatively hard to look at. Now, I'm going to change tack for a moment. So the last thing that I've been asked to talk about is one of the more challenging epigenetic disorders. And this is called pseudohypoparathyroidism. So I want to talk about the new principles that have been developed or thinking about this. And basically I'm going to cite the consensus statement that's just been developed on the management of pseudohypoparathyroidism and its related disorders. This consensus meeting was held this year in March, and I am citing the work that was led by Professor Agnès Linglar in Paris. So she sent me these slides and I want to represent them to you because this is going to be published this year and it is going to change the practice or change the way we think about these disorders. So firstly, pseudohypoparathyroidism, PHP, is a classical imprinting disorder. It's a classic syndrome with classic features. You know, it's a clinical description. And it was described in 1942 by Professor Albright. And what he was talking about was children who had a particular set of features. Firstly, they had Albright hereditary osteodystrophy, so they had particular sort of bony malformations. They have shortened fingers, short metacarpals, and they have resistance to thyroid-stimulating hormone. They can have cognitive impairment. They tend to have truncal obesity, which is actually quite a feature in imprinting disorders. These children also had hyperphosphatemia and hypocalcemia. It looked as if they were deficient in parathyroid hormone, but they'd actually got parathyroid hormone. It could be analysed in their bloodstream. And if you give them a great dose of parathyroid hormone, they do nothing. And so it's not that they've got hypoparathyroidism, they've got pseudohypoparathyroidism. So that's why the name was coined. It's a bit of a strange name. It's always been a bit of a bar to people getting happy about this syndrome. Now, this also could be a bar to people feeling happy about this syndrome, really. The gene that is the critical feature for classical pseudohypoparathyroidism is one of the trimeric G protein alpha subunits. This is the stimulating alpha subunit of the trimeric G protein. Trimeric G proteins are second messenger producers in the cell. So there's going to be a stimulus from outside. Second messenger, a G protein, is going to do some signaling. The beta gamma subunits do something. The alpha subunits do something. There is then a cascade of signaling. And what I'm saying, actually, is that PHP is one disorder somewhere on this pathway. Actually, there are multiple genes on this pathway. There are multiple proteins on this pathway. And mutations in these proteins cause a collection of related phenotypes, including, I'm not even actually going to try and say some of these, but there are various syndromes involving bony malformations, mental problems, and hormonal dysfunctions. I'm going to focus, there's been a suggestion in the literature, which is to stop calling these things names like pseudohypoparathyroidism, because it's relatively unclear, and a suggestion to call them inactivating pseudohypoparathyroidism, PTH and PTH RP signaling disorders, IPPSD. This will be more rational, although it's still quite hard to say. Now, so the range of these disorders includes classical PHP, which is these obesity, brachydactyly, and certain clinical chemical features. There's also features only affecting bones themselves, and there are features here which have their own particular clinical manifestations which are related. So these are all related gene disorders causing related phenotypes. Multiple causes, overlapping phenotypes, overlapping molecular mechanisms affecting this one pathway. But I want to focus in on PHP, pseudohypoparathyroidism, because that's the classic gene in this pathway. The others are really quite rare, and this is the imprinted one. So there's no doubt that anyone who starts talking about the GNAS locus is going to scare people. The GNAS locus codes for this alpha protein, the trimeric G protein. It's a long locus. There are multiple transcripts. The one you need to focus on here is the GSA transcript here. The point is, it's actually expressed biallelically in most tissues. It's monoallelically expressed only in renal tubules, thyroid, gonad, pituitary. It's maternal only. So it's an unusual imprinted gene. This is the main reason why these phenotypes are a little bit complicated to look at. Okay? So let's just walk through this. If you have a mutation in your GNAS gene, then effectively you've got half the normal GNAS you should have in most tissues. In those tissues that only express it for the maternal allele, then you've just lost all of your protein. You've lost all of your gene function. That means that you're liable to have the physical features, the general features of Albright hereditary osteodystrophy. You'll have all those bony manifestations. You'll also have the biochemical features. This should mean you'll have classical PHP1A. If you've got a paternal mutation, then you've got a general haploinsufficiency of GS alpha. That means you're going to have the physical features of AHO. But in those tissues where it's only maternally expressed, there's no problem. And therefore, you don't have the biochemical features. And that leads to something called pseudohypoparathyroidism. If you've got one of these two disorders, paternal uniparental disomy or an imprinting error, in most somatic tissues, well, there's no mutation, there's no problem. But the effect in the maternal only tissues, like the kidney and the thyroid, there is complete loss of activity of this protein. Therefore, you don't have the physical features, but you have the biochemical ones. Therefore, what you would expect to see is something called PHP1B, where the children look perfectly healthy, but they've got hypophosphatemia, hypocalcemia. So that's what you're expecting to see. This is quite rational, really, and it simply arises from the fact that this gene GS alpha is only expressed maternally in a subset of tissues. Unluckily, the situation is a little bit more complex than that, which is that, in fact, there is an overlap between the different phenotypes and genotypes of these disorders. And this is just an absolutely recurring feature of everything that you're going to hear in this conference, I think, not just about imprinting, but everywhere else, that when we look at the clinical features, we come to clinical diagnoses. When we look at the clinical and the molecular features, we see that the neat clinical boxes we've been putting our patients into, they don't fit anymore. There's always going to be Gaussian curves, strange outliers, modifiers, and we just need to be very prepared for this, and it's going to be completely general as molecular genomics improves and improves in our understanding. So what I'm saying is that actually if you've got a child with, for example, AHO, there's no reason only to do sequencing of GNAS. You might need to check for a different anomaly. If you've got a child who's got no bony manifestations or physical features of AHO at all, you might still need to do gene sequencing. So you might just have to do wider testing than you're expecting in order to get the molecular diagnosis. Now, this is one of the slides from the consensus document, and this, I must admit, when I saw it, I was kind of surprised. This is from the PHP consensus meeting. This is the recommended pathway for genetic analysis, molecular diagnosis of PHP. And I think while this is an accurate representation, I think it's kind of complicated. And so I've just boiled it down a little bit. So if you want to molecularly diagnose a child who's got hypophosphatemia and hypocalcemia and who may or may not have features of AHO, then this is what you need to do. You need to combine gene sequencing and imprinting analysis, and then depending on the results, you will get a different molecular diagnosis. And so I think this is broadly how this disorder should be treated molecularly. In terms of clinical treatment, this is the recommendation of the consensus. The consensus is being written now, the meeting is being held. It normally takes about 6 to 12 months to get the document out. And this recommends the different management interventions that should be performed either simply at diagnosis, for example, genetic counselling, offering family support, and the different things that should be done as the child grows older into adolescence and then into adulthood. And so those will be the recommendations of the consensus. Now, I feel I've gone on quite a long journey here, so I just want to summarise this. Firstly, as I say, the principles of imprinting are easy to understand. Some DNA sequences escape reprogramming in the early embryo. They are there and imprinted. Whether these actually cause syndromes, that's a different question. Imprinting disorders are caused by altered expression of imprinted genes, however that happens. It can be a mutation, it can be a deletion, it can be an imprinting change, it can be a uniparental disomy. Imprinting disorders are epigenetic syndromes affecting growth, development, metabolism, and actually behaviour. The features can be a little bit complicated. There's clinical heterogeneity, clinical overlap, mosaicism, and not to forget multilocus imprinting disorder, which can have a range of genetic and epigenetic causes. And finally, I just wanted to keep stressing, classical clinical syndromic definitions should be broadened by the use of molecular diagnosis. I've just presented a little bit about PHP as one example of this. And that's where I leave it. Thank you.